We've got a great speaker for today, all the way from Slovenia and originally from Portugal, and it's Dr. Teresa Oliveira. I have trouble with the last name. And she's currently working at a university in Slovenia. She's part of a big collaborative network in Europe studying Eurasian lynx, and she's going to talk about that work today. But I think it's exciting both because lynx are pretty exciting, but also exciting because of the scale at which they're working, and they're combining data from multiple studies across pretty big portions of Europe. So I think, yeah, really interesting in that regard. Looking at lynx ecology and predator-prey interactions with implications for both basic ecology and applied ecology. So I got to know Teresa actually, I think, when she was doing her master's. So she did her master's at the University of Porto, studying wildcats, and I think initially emailed me with some questions about spatial ecology, and we started kind of going back and forth. And it actually ended up, she introduced me to some folks that I began collaborating with studying wolves. In Portugal, they invited me over, and I got to meet Teresa in person. We stayed in touch. We've published a paper together. Yeah, and just had a lot of common interest in carnivore ecology. So it was exciting when she wanted to come over and spend some time in our lab. And so she's been here for a week, and she'll be here for the rest of this week. And I asked her to do this seminar. And so, yeah, hopefully everyone will enjoy it. I'm sure there'll be some exciting stuff. And without further ado, I'll turn it over to Teresa.
Yeah, okay, yes, no, okay. So can you all hear me? Okay, great. If not, then let me know and I'll speak a bit louder, I guess. So yeah, I'm going to talk to you today about some of the work that we are doing with Eurasian lynx in Europe, mostly about foraging ecology. And I'll start with a few facts about the Eurasian lynx. So the Eurasian lynx is a medium-sized carnivore that weighs roughly 20 kilos, with differences between males and females. The males are usually bigger, roughly 25, 26 kilos, and the females are 17, 18 kilos. And this is the largest of all the 4 lynx species. So the Canada lynx, Siberian lynx, and bobcat are all smaller than the Eurasian lynx. They are seasonal breeders. They have 1 litter per year, roughly 2 to 3 kittens per litter, and they give birth at the beginning, middle of May. And then the kittens start dispersing a bit before they complete 1 year old. The Eurasian lynx has one of the largest distribution range of the cat species. So the distribution goes from somewhere in France all the way to Eastern Russia, which is quite impressive. And also in terms of latitude, you can see that the range is also quite big. In Europe, focusing now more a bit in Europe, they are associated with forested areas in most of the populations. And they also act as apex predators. And they are specialized in ungulates, which makes them, again, different from all the other lynx species, which are mostly specialized in smaller prey, for example, birds and lagomorphs as well. And now, before I go more into the actual foraging ecology, I would just like to give you a bit of an overview of what's happening with the lynx populations in Europe. So zooming in a little bit. This is more or less the current distribution of lynx in Europe. It's a bit outdated, but not really that much. And you can see here that the populations in Central Europe are actually quite scattered and a bit fragmented. And these are populations of conservation concern. The only populations or the populations in Europe that are autochthonous and that don't need any conservation effort, let's say, is the populations in Scandinavia, so all the area in Sweden, Norway, and Finland, the Baltic population in Estonia, Latvia, and so on, and then the Carpathian population, which is mostly in Romania, but goes also to Ukraine and Slovakia. And then we have the Balkan population in the south, which is also autochthonous, but it has some conservation efforts right now. And zooming in a little bit more into the area where I'm actually working, and I'll tell you a bit what happened there in terms of conservation, which is actually then similar to what's happening in the rest of Central Europe. So focusing now in the Dinaric Mountains and the Southeastern Alps. So starting with the Southeastern Alps, which are basically located in North Slovenia, so the upper part of that circle there. And you can see that this area, it has the classic alpine setting, so high elevation, very rocky, low vegetation density, snow most of the year. And then we have the Dinaric Mountains, which are right below the Southeastern Alps, and they go from central Slovenia all the way down to the Balkans to Montenegro, yeah. And the Dinaric Mountains they're really pretty. So it's this kind of karstic landscape with very dense forest, very hilly, without many peaks, but there are still some peaks across the whole range. The forest is very dense. It's full of very high density of different prey species, high density of large carnivores, bears, wolves, and lynx. And it's basically the perfect habitat for most of these species. But in the Dinaric Mountains currently, or at least up to a few years ago, the population was actually in danger because of inbreeding. And what happened there is that the lynx was basically extinct and didn't exist in the Dinaric Mountains until up to the 50s, 60s. And then in the 70s, in the beginning of the 70s, a group of hunters thought that the lynx is supposed to be in the Dinaric Mountains. They are part of the landscape. So they thought, okay, let's reintroduce lynx. So they brought 6 individuals from Slovakia, from the Carpathian Mountains, and they released them in Slovenia. And then these individuals basically created the whole population. So the population increased a lot. There was a huge expansion. They went up in the Alps. They went down south in the Dinaric Mountains. But as you can imagine, a few decades later, they started realizing that the population was declining and something was wrong, at least with some individuals. And this was kind of expected because of inbreeding, because the whole population came from 6 individuals. And in 2013, our team published a paper basically confirming the inbreeding and actually then basically trying to decide what to do next. And as you can see in this picture, this was a lynx captured a few years ago, and he has like 2-year tufts, which is not normal. So yeah, this is one of the signs. It's not that problematic actually for the individual, but they had also heart problems, skeleton malformations, and so on. And so the solution to save this population was then population reinforcement, so bringing individuals from other populations. And then a project started, the Life Lynx, where the idea was basically bringing individuals from Romania and Slovakia to Slovenia and Croatia, so to the Dinaric Mountains. And this project had 2 phases. The first one was to translocate individuals directly into the Dinaric Mountains to reverse population decline and to reduce inbreeding. And the second phase was a translocation of individuals to the Southeastern Alps, so the first picture I showed you before. And the idea was to create a stepping stone population that would connect the Dinaric Mountains to the Alpine population. And this project was quite successful. There were many positive results, but in terms of inbreeding, you can see in this picture, so if you look only at the brown line, this is what would happen to the inbreeding of the population if there were no lynx translocated. So the inbreeding would continue increasing. And then around the time when the lines separated, it's when the reintroductions happened, roughly. And you see that the inbreeding, counting not just for the remnant individuals, but also with the reinforcement. So the yellow line, you see that the inbreeding decreases a lot. And when the alpine stepping stone population is included, then the inbreeding reduces even more. So this was actually a very positive story. And what happened in the Dinaric Mountains happened in other populations across Europe. For example, the alpine population in South Germany and so on. But the thing is, even though this is quite, it's effective, it seems to work, right? Releasing individuals, it's not a viable, it's not something that can happen all the time. We can't just keep bringing individuals every 5 years or every 10 years. So the idea is actually that a viable metapopulation of the Carpathian lynx would be created in Central Europe. So this is what it would look like ideally. So in red, you have the Dinaric population that I've been talking about. The stepping stone is the orange part. And the idea is then that the stepping stone population connects to the Alpine population in yellow and also to South Germany. And that these populations are then eventually connected to the Carpathian and so on, and also with the Balkan population. The thing is, this is not that simple because Europe is quite fragmented. The habitats in a lot of places are really destroyed. There are very big valleys, large industrial areas, so highways. So it's not that easy to have this connectivity working. For example, even just with the stepping stone and the Dinaric population, it seems that it would be quite easy for them to be connected, but there is quite a big highway there that prevents a lot of individuals to just cross. But I mean, hopefully this will work, in the future. So at the same time that all these introductions happened, or even a bit before, there was a group of researchers that wrote key actions for conservation and management of all the large carnivore species in Europe. And this document includes specific actions for all the species. So for example, what can be done in general across all populations, for example, establishing a systematic monitoring, but also then key actions that would be population specific and also across all populations. And one of the actions that was proposed for the lynx across Europe, so for all populations, was actually the integration of lynx predation impact into wildlife management practice. And then you could be wondering, so why would this be important? Why would we have to study predation? Why would we have to incorporate predation in this? So predation is basically, it's an essential ecological process where you basically have a predator looking for prey, capturing, consuming it, and then repeating the whole thing over and over again. And predation can be measured with a few parameters that can be indicative first of the energy that the predator is expending, which is then, or better, that he's obtaining, which is then directly influencing the predator's survival. So it's important for the predator's perspective, but also from the prey perspective. So measuring these parameters allows us to understand how the prey population dynamics vary and how they are affected by predation. And this was studied already, not just a bit for lynx, not that much actually, mostly for wolves and mountain lions. And for lynx, this is important because actually one of the main problems in conservation with lynx right now across Europe is the perceived or real competition with hunters. So basically hunters think that, or a lot of people would say that, oh, the lynx is in my area, so he's going to hunt all the deer, and then we don't have any more deer, the deer will go extinct. And so the idea is then to understand better how foraging ecology, how predation works with the lynx, and then communicate this properly, and also eventually integrating this into management plans of prey species. And as you can imagine, there are a lot of studies for lynx about this, but they are all at a local scale. So they have relatively few individuals. There aren't many environmental variables included because there is low variability within the populations. And that's when we then started working on lynx foraging ecology across Europe. And we obtained the data because you can imagine we didn't, just one group wouldn't collect all the data. We had to collect information from many study areas. And so basically this was provided by the Eurolynx network, which is a network of data sharing, and basically a research group provides the data that they have available. The data is stored in a database. And then any researcher can say, oh, I'm interested in studying, I don't know, variation of home range across Europe. And then the people would be, and then the researchers would be like, okay, use our data or don't use our data. So anyway, then we collected data through this network, and we studied two main topics. The first one was then the variation of foraging parameters across Europe. And by foraging parameters, we considered interkill interval and handling times. And I'll explain a bit later what that is. And also the spatial distribution of kill sites during the denning season, which is when a female has the kittens and they are immobile in the den. And now going back again to to Lynx ecology, but now just to the prey preferences, because this is important to understand the foraging ecology. So I said before that the lynx is specialized in ungulates, which means that they kill prey that it's the same size of the lynx or bigger, often bigger. And basically, in terms of main species, so ungulates, they hunt the roe deer, which is this first deer here. It's not that big, but it's still around 25. It can get to 30 kilos or even a bit more. So it's still bigger than the lynx. Then we have the alpine chamois, which is not as important. It's more important in specific areas where they exist, which they are more connected to rocky areas, for example, the Alps. And also the red deer, which is this deer then here on the left. This one is quite big. It's kind of like the elk that you have here, a bit smaller still, but it's quite large. And the lynx can hunt these animals, mostly females or juveniles, so they don't really go for these big males. But still already the females are quite large. And besides the ungulate community, they are also really dependent on small prey. So hares, also different species of birds and rodents. And this varies also across populations. So for example, in the Balkan population, they really rely a lot on hares. While, for example, in Central Scandinavia, they rely a lot on these birds, on the capercaillie and also other smaller prey species. And then to study foraging ecology, especially of carnivore that hunts ungulates, we needed to then detect these kill sites. We needed to see where the lynx was actually hunting the prey, finding it, and we wanted to measure how long would they take feeding and so on. And for this, we had first to capture and color the lynx. And then we would get from the GPS collar, we would get the GPS data, which could look something like this. And then we would look for GPS location clusters, which would be clusters of points, which indicates that the lynx was stationary for a couple of days, which to us, it was indicative of a prey, of having a kill, because if they then take a couple of days to feed on the deer, because it's quite large, we would detect that from the GPS data. And then we would have to go in the field and see, okay, is it a kill or not? And in this case, for example, it was. You can see the lynx there and the deer a bit below. And then we were actually wondering at some point, and can we actually compare the kill sites data that we get across all these populations? Because we got data from all those different groups, but we were thinking, okay, but they actually have different methodologies. They have different color schedules. They have different field efforts. So they probably detected the kills in a different way. And they also went in the field at different rhythms. Some groups would go really often for like a month. Others would go more opportunistically. So no, we couldn't actually compare this data directly across populations. And we had to work on a method to actually create a comparable data set across all these populations. And so the first questions that we asked there was, how many GPS fixes do we need to detect these kill sites? So not to find that we would go in the field and find them, but that the GPS cluster was generated depending on the number of GPS fixes. And here we use 3 different areas at first. So we included data from North Norway, so all these areas had different prey systems basically. And in North Norway, they had the semi-domestic reindeer, which then, because they are domestic species, then the lynx behaves a bit differently at the kill sites there. And then we had the second study area, which had a mix a bit of everything. There was semi-domestic reindeer, there was domestic sheep, there was a roe deer. So it was quite diverse as well and different behaviors. And then we had the population in the alpine population in Switzerland, which had mostly just wild prey. So roe deer and chamois. And then when what we did is that we got all the GPS data from that we got, and we started subsampling all the data, and we saw when do we stop detecting these clusters. And then we measured like this the success rate. So at first we realized, okay, if we reduce the number of fixes, the success rate would be reduced. So we detect less kill sites, which makes sense because we know that then the cluster wouldn't really be generated. We also saw that using the higher number of fixes per night would increase the success rate, which also makes sense because we know that the lynx are nocturnal or crepuscular, so they would feed mostly during the night. We saw that including domestic prey and small prey would reduce the success rates. And basically, this is connected to the fact that with small prey, the lynx would feed faster. So maybe they would stay half a day or less, or just a bit more, but they would stay less time than they would spend in an ungulate kill. So if we reduce the number of fixes, we don't detect this prey that often. And with domestic prey, they had also a bit of a different behavior there, which affected the way these GPS locations were distributed. And to answer the question, how many fixes do we need? We needed 3 fixes per night to detect 95% of these clusters. So then we were like, okay, fine. We know how many fixes we need to generate these clusters, but not all of these clusters would be kill sites. So we were wondering as well, can we predict which cluster is a kill site or not across all these populations? And at first, so we used the random forest algorithm, and we tried first to compare. We tried a multi-class classification. So we tried detecting clusters that would be ungulates against clusters that would be small prey and non-kills. And ungulates here, when I mention ungulates, I'm referring to adult individuals, so individuals that would be more than 7, 10 kilos, while small prey is then lagomorphs, birds, and so on, but also smaller deer, so neonates. And non-kills would be, for example, resting sites. And this didn't work that well. You can see that the accuracy was not that high, and this was because the small prey were actually misclassified as non-kills. So then we thought, okay, what if we put small prey and non-kills together and create a binary classification? So ungulates against all the other clusters. And this worked quite well. We got an accuracy of, on average, like 85, 86%, which was then really good news. And we used then this model to create our comparable data set. And now moving then to the variation of these parameters across Europe with the standardized data set that we created with the previous work. This was, as you can imagine, a very collaborative, highly collaborative work. We have this paper now finally accepted. It had almost 40 co-authors because we had data from everywhere, and there were a lot of people contributing to this. And so here we used then data from more than 100 links. We created our predictive model with 3,500 or almost clusters. And then we predicted from this model almost 4,500 ungulate kill sites, adult ungulate, and we use this to estimate interkill interval and handling times. And so interkill interval is basically the time that passes from the start on one kill minus the start of the first kill. And it's composed by 2 parameters, the handling time and the searching time. And the handling time is the time that the lynx spends feeding on a kill, or not just necessarily feeding, but just handling the prey. And searching time is the time that passes from the lynx or the predator abandoning the cluster and starting a new one. And so here we focused on the interkill interval and handling times. And also it's important maybe to say you might be more familiar with the term kill rates and not interkill interval, but basically when we have then longer interkill intervals, it means lower kill rates because longer interkill interval means that the predator spent more time finding another prey, which is then reflected in lower kill rates. And then at first we looked at the variation of these parameters across populations, and I'll not go a lot into detail here in each population because it's not that important. I just wanted to point out that the population that had a longer interkill interval was the Balkan population, so the third. And you can see that, if you remember what I said before, that the Balkan population relies a lot on small prey. And this was then reflected in these lower kill rates of adult ungulates. So it was actually nice to see that these results, when we compiled all the data, that they reflected what we knew from different populations. And then we also looked at the variation of interkill interval, but across social status. So we have females with a longer interkill interval, which means then lower kill rates, followed by males and followed by family groups, which is something that we would also expect. And this was consistent across populations, and it confirms previous results, not just on lynx, but also other species and other felids. And when we would look then at the variation of handling times across populations, again, here I'll not go into detail. I would like to point out that you can see that the variation across populations is just much lower. So they are relatively similar. And if you remember from what I said before, the interkid interval is the sum of handling time with searching time. So if you see that the handling time is not varying much, what's influencing mostly the interkid interval is actually the searching time, which is the time that then the predator takes to find another prey. And this is then mostly correlated, as you can imagine, with prey availability. If there is more prey, they would find probably the prey faster, and so this searching time would be reduced. And looking at the variation of handling time across social status, we have again a similar trend. So the females stay longer at the kill, so they have a longer handling time, followed by the adult males, and then by the family groups. So the family groups are female with kittens, and they would take less time eating because they have higher energetic demands, and it's anyway more individuals feeding in one deer. And I actually have an example here of a female with 3 kittens, and you will see how they are not very happy with sharing the food. So this is the mother, obviously, with a collar. So this is the kitten already complaining. And wait for it. Sorry. So yeah, they're not willing to share a lot of food, and you can imagine that these 4 individuals in 1 or 2 nights maximum, they can eat food here easily. Still not happy. Okay. Then we looked at the variation of the interkid interval also along the year, because we would expect some seasonal variations here. And indeed, we found then some patterns here. So we saw that there was a shorter interkid interval at the end of the winter, which means they had higher kill rates. And this makes sense because this is the time of the year when the deer are in poorer physical condition because they had lower food availability in general during the winter. And we think they might be more, how can I say? So the lynx can capture them more easily, let's say. And then we have a peak in the summer. So we have longer interkid intervals, which means lower kill rates. And it's also in the summer when there is in general higher availability of small prey, also including the neonates. So that's when the deer are born, and the lynx also feed quite often on the small deer. So this was also according to what we would expect. And then we were also interested in seeing if this variation was just the same across populations or if there were some different fluctuations. And there were indeed, and quite interesting ones. And again, I'll not go through all the populations, so I'll just show here 2 cases. So we have again the Balkan population, which is the one with the light green, the curve that's more, in the top there. And you see that they have a very strong variation of interkid interval along the year, which means that during the winter they were hunting more deer. But then in the summer, they almost completely shift and they consume a lot of small prey because they have very long interkid intervals of adult ungulates. And in contrast, we have the Carpathian population, so the red line, which you see that it's almost flat, which means that there were no variations along the year. The lynx was basically hunting adult deer all year long, which indicates basically a very good prey basis in that population. And then we also wanted to look at the variation of these parameters, but in terms of some environmental factors, because we had such a high diversity of environmental factors that it would be good to test this as well. And as you can imagine, the first thing that comes to mind that can influence these parameters is prey availability. We didn't have, unfortunately, comparable data on, for example, deer density across the population, so we had to use proxies. And the proxy that worked better was environmental productivity, measured as NDVI. So basically, where we would have higher levels of productivity, so higher values of NDVI, this would be reflected or would mean higher prey density, so more deer. And if we would have lower productivity, this would be reflected in lower prey density. And here we look more on the right side of the plot, because it's where we had more data. The first, in the left side, we have relatively few data points. And here we see that when we have less productivity, we have a longer interkill interval, which means lower kill rates. So low productivity, lower kill rates, and then higher kill rates in areas that we have higher productivity. And this pattern was actually similar across populations. So that was interesting. And then we looked at the effects of scavengers' presence. And here we consider just the dominant scavengers, so not the whole scavenger species, but we focused more on the presence of brown bear and the wild boar. And we saw that the presence of bear and also the presence of wild boar would actually reduce the handling times. So wherever there would be bear or wild boar present, the lynx would stay less at the kill, less time. And in terms of kill rates and interkill intervals, we saw that the presence of bear didn't really affect the interkill interval, but the presence of wild boar would then reduce the interkill interval. So they would, the lynx would increase the kill rates when the wild boar was present. And we know that the scavengers have a very strong impact on lynx kill rates. We know this from previous local studies, and we also know that scavengers in general affect kill rates in other species. But here, especially in this study, I think we don't really get the whole picture, because I think we still missed quite some kills, because it happened relatively often, at least in the Dinaric mountains, that the bear would come and take the deer in the first night or the first couple of nights, which then means that, you know, probably our method of classification wouldn't really classify these clusters as kills. And I'll show you here an example of a bear coming and stealing the whole thing, the whole carcass. So here you have, yeah, the quality is not that great, but I think you understand. They have the lynx feeding. And then she's also covering the carcass, which is something that we think they do precisely to avoid scavengers, so that the smell doesn't really travel that far. But here didn't really work that well. You will see that she then becomes aware that something is wrong. She is there. So then you can imagine the lynx really can't do anything with a bear like this, so they just simply move away. And then the female came with 2 cubs, so there's even less food. So, I mean, less chances that anything is left. And they either just feed there on the spot or they move the deer. And then, yeah, usually the lynx comes back and realizes that there's nothing there left. And then they'll have to get a new prey soon. And another example here, we have in this case a male, and again he feels that, yeah, there's something coming, and he just goes away. It's better safe than sorry, right? And then a big bear family comes, so not just a mother with 1 or 2 cubs, but she comes with 4 cubs. So it's suddenly 5 bears eating a small deer. So again, as you can imagine, there is really nothing left, and they, again, either they feed on the spot or they drag the carcass, but they don't leave anything, maybe just the spine in the end. Yeah, and then again, the lynx, after the bears are gone, the lynx usually comes back. Where is he? He will come eventually. And then, yeah, he comes back and checks and sees, yeah, there's nothing left. And they'll have to get another prey soon. So yeah, I mean, of course, this depends as well a lot on the density of scavengers in the area, and all these cases are in the Dinaric mountains, where we know that there are a lot of bears everywhere, but it's still a very strong influence on the kill rates of the lynx. And now the last parameter that we measured was human presence. We also expected that human presence would affect these kill rates and the foraging parameters. And we looked at the effect of human presence actually in terms of the feeding time, and we used here 2 variables. We used the human modification index and the distance to settlements. And we see basically where there is less disturbance, we have a longer feeding time, and when we have more disturbance, there is a shorter feeding time. So when we have more humans, more cars, more higher settlement density, the lynx doesn't stay that often, doesn't stay that long in the queue. And when there is less disturbance, they stay a bit longer. And so this was a relatively clear trend, but then we also wanted to check how, if this would vary also among populations. And it did. And it was actually interesting because we saw that in the populations where there was higher human, human disturbance, let's say, the trend was the same as before. So it was quite, with less disturbance, they would feed for longer. And then with more disturbance, they would feed for a reduced amount of time. But the populations that had in general lower human density, disturbance, for example, the alpine, like the blue line flat, you see then that the human disturbance doesn't really affect in general the handling times. So then we see different levels of sensitivity from the lynx with then different gradients of disturbance. And now just a few take-home messages for this part. So we see a high adaptability to human impact from the lynx. So when there is higher disturbance, they are also more sensitive. We see also that the main negative impact of human presence in general to lynx foraging ecology is related to the predator's energy uptake. So so basically when there is more humans, they reduce the consumption of prey, which then makes them having more need to hunt more regularly. And this was found already before locally in a few lynx studies, but also in mountain lions. And we also see from this study that understanding these variations across such a range is crucial to then implement particular effective management measures then on the population level. One thing that we didn't check here, and it would be interesting, is potential cumulative effects. So we checked these factors individually, but we also think that, for example, if we have areas with high human disturbance and then a lot of scavengers, so high scavengers density, this could then have an even stronger impact on these parameters. And this is something that would be interesting to check in future studies. And regarding that, so what's next in this topic? We are actually now applying for a project where we want to use activity data, so accelerometer data, to actually detect particular behaviors of lynx. We are mostly interested in feeding, because then we think that if we detect feeding, we might be able then to detect ungulate kills that had a very short handling time. So for example, if the bear came and took the carcass, or if the lynx abandoned the prey because there were a lot of humans around, we will still detect this kill because we detect the feeding. So we wouldn't rely on the cluster or on the GPS data. And this would then have direct implications for management, because we can actually then adjust the hunting quotas. And this was done actually for this paper, which used the Canada lynx. And yeah, ideally we can do something similar, even though they managed to detect small prey. And ideally we would do that as well, but I don't know if that will work out. We'll see. You see here, this is a picture of a lynx from Romania with a squirrel, which is something we really don't see often. Not in the cameras, not even in kill sites or anything. So it's yeah. Hopefully we get more data on this. And now moving to the spatial distribution of kill sites during the denning season, which is the second topic I mentioned before. Here we again use data from some populations across Europe with quite some latitudinal range. This work was actually conducted by a master's student named Alpia. She did a really good job handling all the data. And most of her thesis and also the paper that we are working on now, it's focused on movement patterns. But there is also a component of prey use, which is what I'm going to show you today. And in case you are not familiar with this, so this would be what the GPS data would look from a female that started denning. So in the circle in the center, you would have the den or the denning area where she would move the kittens a bit around. And then she would have some kill sites around. So this would be then the squares, the red squares. And you see that the female goes back and forth often. So she's at the den, she goes to feed, and then she comes back to the den and goes back to feed, and so on. And then this creates this kind of like star-shaped movement pattern. And actually these kittens were actually found in this particular den of this female, which is also what I'm going to show you here in this video. So you see that after we went there, the female was not there. Yeah, they're very loud. So, and they're very happy that the mother arrived. So she goes there, she stays with them a bit. And they're resting and she's feeding them. And then eventually she starts moving them around. So often they would move long distances between dens, but also in a given area, they just stay, they just move, let's say 100, 200 meters, and they just go around the area, which is also a defense against scavengers. So you see that she starts, carrying them and moving. This was actually quite an operation because she had 4 kittens. And it was a drama at some point because they all start crying and falling and rolling, and then she comes back. Yeah, you can hear them in the back. And then she comes back and drops one, gets the crying one, and yeah. Anyway, but she managed quite well, and all the 4 kittens survived up to the dispersal age. So yeah, and then one thing that we know already from this study that we are working on is that there are differences in daily distances between regions. So in Northern Europe, where it's more cold and there is lower prey density in general, the females would move much longer, daily, when compared to, for example, Central Europe, where there is much more prey availability. And yeah, we think it's connected to prey availability. Again, we didn't have a good layer to test this, but at least it matches our predictions. And in general, there is very little information about the distribution of kill sites during this period, and precisely because this is such hard data to get. And then if we had all the different research groups with the 1 or 2 individuals, it's not really easy to do any comparison, let's say. But thanks to this network that everyone could put all the data together, we could then actually look a bit more into detail into this. So we had a few hypotheses here. We first expected that the kills would be distributed around, let's say, within a belt around the den. So they wouldn't be too close to the den, because then there are higher chances of attracting scavengers, especially if it would be like a deer, because they could attract then the bears, foxes, and so on. But they also wouldn't be that far away, because then that would mean that the female would have to walk for quite some long distances, and that would be not very efficient energetically for her. And this would also mean that she would have to leave the kittens unattended for longer, and they need to be fed. They need help with thermoregulation, so it's not that good if the female stays away for that long. And then we also expected that the distances between a kill and the den would be longer in the northern areas, which would be then connected to this lower prey availability. And we also predicted that kills of small prey would be located closer to the den, precisely because there are lower chances of attracting scavengers because they would eat faster. And then here's all the data that we used, and we split this basically in 2 regions: the cold region, which would be central Scandinavia to the north, and then the temperate region, which would be more Central Europe and South Scandinavia, where the prey species, the density is a bit larger, and they anyway mostly feed on roe deer. And then what we had here, basically, we had, let's say, our den site, and then we saw indeed that, okay, there was some variation in the distances to the den, but we see that the closest kill site was actually located roughly on average, or a median of 1 kilometer of distance, and the furthest kill site was 3 kilometers. And so we do see that there is some kind of donut shape, let's say, distribution of kill sites. But since we also didn't have all the kill sites confirmed, we couldn't really look a lot into detail into the real spatial distribution, because we don't have the full data set of all the prey that the female got. But this is already a quite interesting result, and that tells us something a bit more of this behavioral adaptation of the female during denning. And we saw also that small prey kill sites were then located closer to the den, so there was a significant difference between small prey and ungulate prey, which was according to our expectations. We didn't find a significant difference in the distances of all kill sites, but in compared to northern areas and the north area and the temperate, which was not that expected. You can see also in this plot with the raw data that we would still think there would be a significant difference. But in any case, we then looked at the interaction between prey type and the two regions. And we did find that the ungulates were located closer to the den in temperate climates compared to northern climates, which was also what we predicted. And here again, we didn't check all the kill sites, so we are most likely missing small prey, especially because they're really hard to detect. But I believe if we would have everything, we would see, at least in the temperate region, these distances would be much shorter. And now just a few take-home messages for this part here. So these are still the first findings of foraging behavior during the denning season, which is really interesting. And this gives us a better understanding of how the female Eurasian lynx adapt during this first period when the kittens need a lot of care and the mother presence at the den. We also think, or we didn't look into the effects of this behavior in terms of fitness, but we can assume that the energetic demands of female in the northern regions, in the cold region, might be higher, which can then can then be connected to lower fitness. And although we didn't test this, this was found in wolves across North America. So we think this would be actually something quite interesting to look into regarding about the lynx. And so one thing that we would actually be interested in is to check whether the breeding female lynx are influenced by human disturbance. And if so, does this affect just the litter size in general and the kitten survival up to dispersal time? So the kitten survival, I would say, up to the point that they leave the den, but also until dispersal time. So that's more or less it. I would just like to finish with going back to one of the first pictures I showed you with the links to distribution. So most of the studies we are doing right now are focused on Europe. So basically it's square there. And you can see that there's still, most of the links distribution range, we actually don't really know anything of what's happening there. There are very few studies. We have here a picture of the Eurasian lynx that was captured with the Mongolia Snow Leopard project, which my supervisor is part of. And you can see, I mean, they're still majestic, but you can see that there is really nothing around, and it looks very harsh. There is no vegetation. We don't really know, how basically what they're doing there, what they're eating, where they are sleeping. We know nothing. So it would be really cool in the future to also, expand a bit the range of our knowledge about Eurasian lynx. And I finished my presentation with this. Then just obviously there are tons of people to be acknowledged. Not just the research group I work with daily, which are great people and really amazing friends as well. I showed you some results of the Life Lynx project, which, by the way, there are 2 documentaries available online about the project. They are roughly 30 minutes each. If you are interested, I can either send to John and then they can be spread or, but anyway, a lot of people involved in this project, and obviously many people from the Eurolynx network provided not just data, but also a lot of their knowledge about their lynx population and also lynx ecology in general, which was really helpful. And yeah, I'm very grateful to all of them. And thank you all for listening. That's it.
Bring the mic over, that way they can hear you on Zoom. Do you know, has all the subpopulations in Eastern Europe of the lynx been historically disconnected like that, or has human development made that worse? I know you said that there are some highways in between different populations. Did that create the fragmentation, or were they already geographically disconnected?
So the thing is, the lynx was basically extinct from Central Europe in the beginning of the 90s, or even maybe a bit before. They were completely extirpated, along with roe deer, for example. They were also extinct for a while, and also other large carnivore species. So basically, all these populations now in Central Europe are a result of reintroductions. I have here a map I can show you. You can see here, so in the blue you have the reintroduced populations, and autochthonous are the green ones. So yeah, basically, and they all came, the source population was the Carpathian population. So now genetically, all the populations in Central Europe are connected to the Carpathian population. And yeah, but it's still, the highways are not making it any easier, you know, but yeah.
Great presentation, and those videos are really cool. So that's what my question's about. So how did you get the footage around the feeding times? Did you get that with the GPS fixes?
Yeah, so when we see this, the cluster generated, when we see, for example, 2, 3 locations, we already think, okay, maybe there's something. And we go there, we find the prey, and we set cameras, and yeah, and then we usually record, yeah, nice videos. And we try to go immediately at first, because then if you go a bit further in, so like the third night, there are high chances that either they don't even come back, or then we just get the scavengers. And we are also interested in checking when do the scavengers arrive and when do they take the prey. So we really try to go as early as possible, especially also when there are kittens. When we want to record them, we try to go early, because otherwise they just eat everything, and yeah, we don't see them.
So I know you said like the genetics because you brought in initially 6 individuals. Are the genetics still a major concern, or are those fixing themselves now?
I mean, it is getting a bit better with these reintroductions, and you saw that in general, the inbreeding levels are much more reduced and back to like normal levels. But it's something that we can't really, like, we can't rest now, you know. It's just something it has been discussed also during the later project meetings, that, you know, now if nothing is done, then this is going to happen again eventually. You know, the inbreeding is going to increase, and then we need to bring more animals, and that's why we also should try to improve connectivity in general. So a more active approach and not reactive. Yeah, but yeah, they are doing much better, fortunately.
How long funding windows do you get?
For example, this project was 7 years, but this was actually not. It was not a research project. It's a management conservation approach. So it was actually funded by the European Commission, and they don't really, in general, fund research projects. So we kind of try to collect the data in a way that then it can be used for research, which is sometimes a challenge. Because, for example, the idea was that the colors would stay as long as possible in the animal, but for that you have to get a reduced number of locations, which is then for research is not ideal. So there were quite some trade-offs there, but in general, like research projects, we would get 3 years, 4, you know, if we are lucky. So yeah,
That's not very long, is it?
No,
Yeah.
not a lot.
Yeah.
Yeah.